All right, good morning. Still in the nervous system, and actually for the rest of the semester, how little there's left of it. Just 2 weeks, I can't believe it. It seemed to have gone really fast. This is where we left off last time, sort of highlighting the similarities and differences between here, what we're talking about, one nerve cell talking to another nerve cell, comparing it to what I already talked about for chapter 9, where a nerve cell talks to a muscle cell. There was a lot of similarity. We're going to pick it up here now. So we have one neuron releasing neurotransmitters onto another one. The neurotransmitters are chemically gated receptors. They open and ions flow depending on concentration gradients, and the more neurotransmitter gets released, the more of those guys will open and the longer they'll stay open. So the strength of the stimulus really depends on how much neurotransmitter gets released by the first neuron, the presynaptic neuron. And the synapses and all of these nerve endings are chemical, right? Using neurotransmitter. And what's different here between nerve and muscle is that they're not all necessarily excitatory. We can also have inhibitory ones. But the principle of how they function in general is the same. So a neurotransmitter binds to some sort of ion channel, it opens up and ions flow. So let's look at that a little bit more. So here is the example of an excitation where the presynaptic neuron tries to stimulate the postsynaptic neuron, and the graded potential is an excitatory postsynaptic potential. And excitation is always associated with a depolarization of various sizes, right? These are graded potentials, they're various sizes, various strength. And here's just a little blip, a little depolarization. Now these are pretty much sodium-potassium channels, just like what we saw on the neuromuscular junction. So the neurotransmitter binding opens a sodium-potassium channel, one channel for both. And then sodium flows in at a much higher rate than potassium will flow out. There's a steeper gradient for sodium. So sodium influx much higher than potassium out. And this leads to a graded local depolarization, like shown here. This little membrane blip getting less negative inside. And if that spreads all the way to the axon hillock, and if it's still of a certain size, the threshold is actually minus 55 millivolts in nerve cells, then it will trigger an action potential in the axon. And we learned before that these local graded potentials, they decay over distance. So there's a chance that whatever reaches the action hillock is not going to be strong enough to trigger that to do anything. Okay, so that's an excitatory potential. Here's an example of an inhibitory postsynaptic potential where the membrane actually gets more negative inside. It gets hyperpolarized. In these inhibitory postsynaptic potentials, we have different channels. So now the neurotransmitter binding opens a potassium channel for only potassium, or a chloride channel for only chloride ions, or sometimes both. Then you have, if it's a potassium channel, potassium outflux. Or in the case of chloride, we've not talked about the concentration gradient for that one, but chloride will flow in. And both of those events make the inside more negative. So we have a local hyperpolarization, again a graded potential. Like on the graph, so it gets more negative inside. And that also will spread to the axon hillock and make it less likely for a signal to start in the axon. Now, I told you before that it wouldn't be uncommon for a nerve cell in the brain to have 10,000 connections to other cells. So what happens is there sort of a summation of all these inputs a cell might have. So some of the stimuli a cell gets may be excitatory, it may get inhibitory signals. And it basically adds everything up, adds all the excitatory ones, it subtracts all the inhibitory ones, and whatever the net input is at the axon hillock will trigger if that nerve cell starts an action potential itself or not. It's called summation. Summation by the postsynaptic neuron of all graded excitatory postsynaptic potentials and inhibitory postsynaptic potentials. The overall process of this summation can be called neuronal integration. So here it has 4 different examples. The nerve cell being stimulated or inhibited is the yellow guy here in figure 19. And then we have some examples of excitatory stimulation. So if we look at A, over here there's an excitatory neuron in green that stimulates the cell here one time, and then after a little while another time. But the rate of stimulation is pretty low, so that the excitation never really makes it above the threshold at the axon, and it's not really doing anything. It's just not enough stimulation. Remember, signal strength is by just going faster. This is just going too slow, so none of that makes it really to the axon hillock in any significant way to trigger stimulation. So nothing happens, no summation. Now the guy's getting more serious about it. He's starting to stimulate more. Boop, 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 right? A faster rate. Well, now if you do that, then these guys will add up. It will sort of build up intensity. You get more neurotransmitter release right there, stronger depolarization of the membrane. And if it's reaching that threshold at the axon hillock, then it will trigger an action potential. And you can get a similar idea if this one guy is stimulating at a fairly slow rate, but another guy is also now stimulating, and if there's enough of the stimulatory input to the cell, you get an action potential. So here they distinguish this, they call temporal summation because it's just going faster. And here is spatial summation because it's more different ones and different locations that are triggering the cell to do something. And then here we have spatial summation where one guy is exciting, the other is inhibiting. Well, that kind of cancels each other out. So if you have some stimulation, some inhibition, that it cancels. So if the excitatory one now would go faster and faster and faster, then maybe that would overwhelm the inhibition. So it's kind of like math, right? You're just adding, subtracting stimulation and inhibition. Okay, so that's all we'll do on chapter 11. And now we'll move into chapter 12, and I'm going to cut some stuff out here, but we'll see how we do. This I just kind of want to show, to explain why your brain is hollow. You actually sit in here, you have spaces inside your brain that are filled with fluid, but that goes all the way back to how your brain forms. And the brain is brain and spinal cord, the central nervous system, is one of the earliest things to form in the embryo. Before you really have a heart, even this starts to form, and it's in a very primitive form, as you can imagine. There are no nerve cell connections really formed yet, but the earliest brain formation is shown here in figure 1233. All the developmental stuff is always in the back of the chapter, so that's why this figure number is so high, and then we'll skip back to the front in a minute here. The brain and spinal cord basically start by pinching in the tube from the dorsal surface of the embryo. Embryos at that stage don't really look like much. They're just sort of oval areas, and they're actually pretty flat like a pancake. And along what will become the dorsal midline, you get an indentation into the surface right there, as you see in cross-section, that then drops lower and eventually pinches off a tube. So this inside of this tube used to be the same as the outside of your body, right? So that it just sort of rolls in and pinches off a dorsal tube, and that explains why your brain is containing all these spaces that we will talk about next. So it's kind of a strange way of forming a brain. So this is now figure 1, showing you the neural tube, this little longitudinal indentation that pinched off along your dorsal midline, called the neural tube. And the anterior and the rostral end of that neural tube gets elaborated to form your brain. And at first, it forms 3 primary brain vesicles, and colloquially they're called the forebrain, midbrain, and hindbrain. And the scientific terms of those are prosencephalon, mesencephalon, and rhombencephalon. So encephalon is always the same part of the word, and just the first part changes. So prosencephalon is the forebrain, mesencephalon, midbrain, rhombencephalon is the hindbrain. And you start seeing that early on in the embryo, the front end of the neural tube expanding in these 3 regions. And then a little bit later, the forebrain and hindbrain subdivide again, and you have 5 secondary brain vesicles. The forebrain, the prosencephalon, divides into rostral telencephalon, and then right behind it a diencephalon. The mesencephalon stays the same, it doesn't subdivide, and then the hindbrain, the rhombencephalon, divides into the met and myelencephalon. And all these different brain vesicles, these 5 brain vesicles, give rise to particular structures in the adult brain, and here are some that are mentioned here. So the telencephalon, the very front of the brain, this will form what's called the cerebrum or the cerebral hemispheres, where you do all your higher order thinking stuff. That's the convoluted looking bit of the brain. If you've seen the brain in lab, this is all telencephalon. The diencephalon is important because it actually gives rise to your eyes, or most of your eyes, and the big functioning part of your eye, the retina, comes from the brain like that, and outpocketing off the diencephalon, and then some other areas that you'll hear about in AMP2 if you take that, like the hypothalamus, which is important in hormone stimulation with the pituitary gland. So it's an important brain region. Then the mesencephalon and myelencephalon basically form what we call the brainstem. Brainstem. And attached to the metencephalon is the cerebellum, which is sort of like a miniature additional brain stuck to the brainstem back there. Now, the space inside the brain, these are called ventricles. So the fluid-filled spaces inside the brain are called ventricles. And here are the names of the ventricles in those parts of the brain. So in the telencephalon, we have two lateral ventricles. The diencephalon then has the third ventricle. The mesencephalon, the one in there is called the cerebral aqueduct because in life that gets really, really narrow, like a narrow tube. And then met and myelencephalon, there it's called the fourth ventricle. And then when you get into the spinal cord, you have a tiny central canal inside your spinal cord. And all of those spaces persist into adulthood. And there's fluid in there called cerebrospinal fluid that helps shape the brain and nourish the cells in there. So you start out with the brain as a tube, and then the front end of the tube elaborates into these vesicles and grows bigger and bigger. And in humans, we have particular growth of the telencephalon here in this tan color. This is figure 2, forming the cerebral hemispheres, and it sort of overgrows everything else. All the other brain regions, the other 4 vesicles get covered up pretty much by this large telencephalon, this is part of the forebrain. And so you have the telencephalon in beige, and then the diencephalon in this purple color, and then the brainstem in green, and that's the cerebellum attached to the brainstem. And all this, these folds and convolutions on the brain is to pack more surface area into your cranial cavity so that your head doesn't have to get ginormous. And this is something you see in mammals. If you were to look at the reptile brain or a bird brain, it would look kind of like this, almost smooth on the surface. So mammals have really elaborated the forebrain a lot. And then humans and things like dolphins have also really large forebrains, particularly large ones. Here is a view of how all this twisting and turning convolutes the spaces inside the brain. So in figure 3, you see the ventricles, how they are in an adult's brain or at birth. In babies, they're already laid down like this. So you have 2 lateral ventricles, a third ventricle, the aqueduct, and the fourth ventricle. And what's important here is they're filled with cerebrospinal fluid, CSF, cerebrospinal fluid, and they're lined with special cells called ependymal cells, or the lining's called the ependyma. And those are cells that are ciliated. They're not nerve cells, they're support cells. And they circulate that cerebrospinal fluid around. And we'll see that there are a couple of areas where the cerebrospinal fluid can get out of the ventricles of the brain to the outside. where it also is important in protecting the brain from the outside. So there's fluid inside and outside. Sometimes in babies, the ventricles get too large because of an overproduction of cerebrospinal fluid. You may have heard of hydrocephalus or water on the brain. That's an imbalance with making the cerebrospinal fluid in infants before they're born, and we'll get to that. All right, organization of the brain. We talk about gray matter, and gray matter is all the nerve cell bodies, so neuron bodies, the dendrites, and any axons that are not myelinated. They don't have those Schwann cells wrapping them. That's your gray matter. And then white matter is all the myelinated axons, and a lot of times they run in so-called tracts of myelinated axons or axonal tracts. Gray matter versus white matter. So now we'll go over the main brain regions. I will not talk a lot about the brain for time reasons. I'll just sort of give you some basic stuff on them. And we'll start with the forebrain and work our way back. So we'll start at the front. First are the cerebral hemispheres, part of the telencephalon. So the telencephalon, the very front part of the forebrain, forms the cerebral hemispheres. And in humans, this takes up over 80% of your brain, is the telencephalon. As I said, that's really elaborated in mammals and humans and dolphins in particular. Very large forebrains. On the cerebral hemispheres, you have gyri and sulci. Gyri are these raised areas, and the sulci are the grooves between them. Gyri are the ridges, the sulci are the grooves. Singular gyrus or and sulcus. And this is probably just to be able to pack in more brain in a smaller area. Fissures are particularly deep sulci. So here in figure 4 in this example, so this is a sulcus, that's a fissure, it goes deeper in. So fissures are deeper. And they're really deep, these guys. They're named longitudinal and transverse. The longitudinal fissure is between your right and left halves of the brain, longitudinally down the midline. And then the transverse fissure is really between the forebrain and the cerebellum back here. So they're really deep. And then we have some named sulci and some named brain lobes. So we have pretty deep sulci separating 5 named brain lobes. First is the central sulcus that separates the frontal from the parietal lobes, and in figure 4 the lobes are all different colors. So in red is the frontal lobe, and this shade of blue here is the parietal lobe, the central sulcus, separates the frontal from the parietal lobe. Then we have a parieto-occipital sulcus that separates the occipital from the parietal lobes. The parieto-occipital sulcus between the occipital and the parietal lobes. So there's the occipital lobe. It's not really illustrated here. They put a label on there, but it's kind of deep in the sulcus. Then we have the temporal and the frontal lobe separated from the parietal by the lateral sulcus. That one is very prominent along here. So the lateral sulcus separates the temporal and frontal lobes from the parietal lobe. And then we have a fifth brain lobe, the insula, which is kind of in the gap. If you pull the temporal lobe down, you get to the insula. So we have frontal lobe, parietal lobe, occipital lobe that kind of matches with the bones, if you remember. So that way we'll be able to remember that fairly easily if you go back to your bones, right? Frontal, parietal, occipital. Frontal, parietal, occipital. Same temporal bone. Okay? So the only one that's kind of funny is this insula that's kind of deep in. Now, the cerebral cortex we'll talk about a little bit, and then we kind of just hint on the more basic brain structure. So functional areas of the cerebral cortex. The cerebral cortex is the most superficial layer of the forebrain, of the telencephalon of the forebrain. And in it we'll have motor areas, we have sensory areas, and we have so-called association areas. Motor areas, sensory areas, and association areas. Association areas is sort of making sense of stuff. You can think of it that way. Interpretation. We have two hemispheres, a right and the left brain, and they control the opposite sides of the body. Contralateral means on the opposite side. So my left brain operates my right side of the body and vice versa. Hemispheres control contralateral sides of the body. And there's also a little bit of lateralization of cortical function so that each half of the brain has some unique abilities. So the left side tends to be language, math, and logic type of functions, and the right is visual, spatial, intuitive, and artistic. So there's right and left brain differences is what this means. And if you're left-handed, those are probably reversed in you. So the population of left-handed people, those are switched. At least there's pretty good indication that they are. But these areas are not completely independent. So there's a lot of crosstalk in the brain and a lot of overlap. And even the ability to have, if there is damage in one of these functional areas of adjacent areas, to learn some of those functions. And most of this work is from accident victims and stroke victims and brain tumor victims that had parts of their cortex destroyed. And then you know, well, after having a stroke there, what can't you do anymore? So that's where most of this knowledge comes from. Here's a map in figure 6 of the cerebral cortex. The cerebral cortex is actually pretty thin. It's the superficial layer of the telencephalon, and it's only 2 to 4 millimeters thick, and it's all gray matter. And the map shows some of the functional areas. I'm not going to ask you to remember all of these. You can take psych courses if you're interested in this stuff. We're really kind of running out of time, so I don't want to spend too much time on here. Just kind of highlighting a couple. For example, the vision area is actually at the very back of the brain. You might think it would be further forward where the eyes are, but the optic nerves input kind of under here. You don't see it in the midline, and they relay all the visual information to visual cortex area. And you see here the subduet primary visual cortex, where the sensory input goes to, and then there's a visual association area where you're actually making sense that the lines and circles and whatnot that you see might be a bicycle. You know, that kind of stuff. And this is way back in your occipital area, and this is probably why if you get hit in the head back there, you may actually have visual hallucinations, like you're seeing stars if you hit your head, because the only way electrical activity back here gets interpreted is, oh, that must be visual input. So if you have any sort of stimulation of that area, it will be interpreted as vision. The hearing stuff is kind of where the ears are, so that makes a little bit more sense. And you can look at some of the other things. The very forebrain part of the frontal lobe, this is complex stuff. Thinking tasks right there in your forebrain. So executive area for task management, working memory for object recall, solving complex multitask problems. So all our really higher order thinking when you're sitting there taking a physics exam, forebrain, right there in that frontal lobe is where all that is happening. These two I'll focus on a little bit more. Here's that central sulcus that separates the frontal lobe from the parietal lobe, and right in front of the central sulcus there's this big motor area, the primary motor cortex, and then right behind the central sulcus in this darker blue here is the primary sensory cortex, and that has been mapped pretty well here in figure 7. So this primary motor cortex is in red, motor, and you can make a motor map. This is where all your voluntary movements ultimately start. Unless it's some sort of reflex that you stepped on a tack and withdrawn your foot, if any sort of conscious voluntary movements of your skeletal muscles start in this precentral gyrus, that gyrus right in front of that central sulcus. And this funny looking map that's so out of disproportion, they drew the figure on there to illustrate how much brain power is devoted to running those particular parts. So you see that the face, lips, jaw, eyes take up a huge area in the brain because of all our muscles of facial expression running all that fine control over those areas. This is incredibly important for human biology, because a lot of our communication is actually this nonverbal stuff with the face. So we have a lot of fine control over our face. The hand, look at that. Lots of fine control, motor control over our fingers to be able to manipulate objects. So we devote a considerable chunk of brain just to move your hands. And then the larger muscles, the arm, trunk, legs, those are big muscle groups where you don't have, for walking, you just swing your leg back and forth, and there's not a lot of terrible fine control there, and same with your toes. The tongue for speaking and swallowing takes up, for considering its small size, takes up a lot of area as well. And then for sensory, you have an equivalent thing. So where's the sensory input if somebody touches you on the hand or if somebody touches you on the elbow? Sensory input, again, from the face takes up a lot. So any sort of receptors on your face, there are lots and lots of touch receptors on your face that input to that part of the brain. From the hand, also sensory feedback from your hand, lots of touch receptors in your hands. This is how we encounter our environment. And then the rest of the body, pretty minimal comparatively. The feet, again, relatively big because you're walking historically barefoot. You have lots of receptors in your feet to make sure where you're stepping. And the tongue, again, relatively large, and the abdominal organs take up very little space. So this is kind of neat how this matches to how much brain power we dedicate to running those areas of the body. So this is all the gray matter. What's also in the forebrain is white matter. And here's just an illustration of some of the axon tracts with these colored lines. Figure 12.8. So we have white matter also in the forebrain, in the telencephalon. And we have different types of axon tracts. So we have these guys, the association fibers. This is axons that stay kind of within the same general area of the brain for communication. So association fibers close in, short distance. Another type is commissures or commissural fibers. These are connections between the right and left brain to communicate back and forth, right versus left brain. And one of the biggest commissures that you'll see in lab is the corpus callosum. So big right brain, left brain highway for communication. And then the third type are projection fibers. Projection fibers communicate from higher to lower brain centers and vice versa, and even down into the spinal cord. So again, association fibers, local, within the general short area, commissural fibers, right to left, left to right, and projecting fibers up and down from higher brain centers to lower and into the spinal cord. And then lastly, in the telencephalon, we have the basal nuclei, which are gray matter, but they're deeper in the brain compared to the cortex. So here in figure 9, you see this sort of frontal slice through the brain. The darker beige, this is cerebral cortex, then you have white matter. And the basal nuclei here are colored in these reddish and purpley colors. You don't have to know their names. But they are gray matter deeper in the forebrain. The diencephalon is next, still the forebrain. So this is the second part of the forebrain, the diencephalon, the purple bit that's deep in. Parts of the diencephalon are the thalamus. So this is a section of the brain, the thalamus. The thalamus is basically the walls of the diencephalon. So there's thalamus, this general area. Then we have the hypothalamus. Hypo means below, so that's the floor of the diencephalon. And the hypothalamus is there, so at the bottom of the diencephalon, and associated with that area are the pituitary gland, which you'll hear a lot about in AP2, our master endocrine gland, which gets input from the hypothalamus. And we have the optic chiasma entering the brain right there, so that's where the optic nerves attach to the brain. And so those are two important structures in the hypothalamus. And then, just for completion, the epithalamus is the roof of the diencephalon. And one structure there is the pineal gland that you'll see in lab. This thing right there. The pineal gland is associated with your sleep-wake cycle, sort of biorhythm. And then the brain stem is all the stuff in green. Brainstem, midbrain, this part here, medulla. So the brainstem is composed of the mesencephalon. It's the first part of the brainstem, the mesencephalon. I'm just putting these up because I need some picture. There's more stuff on there. You really don't have to know any of these terms right now. At this stage, the mesencephalon is the first part, so that would be the most anterior part of the green area. It contains the cerebral aqueduct. That's the ventricle in there. So it's a really narrow area that still has a connection. Then the next part is right there, the metencephalon. The metencephalon, and on the metencephalon you will be able to identify the pons. The pons is a major relay center where the brain higher centers communicate with lower stuff down the spinal cord. So lots of fiber tracts in there. And this is also, so here's the pons. And this, on the top part of the metencephalon, we will attach the cerebellum. So that's in the metencephalon area. The cerebellum, this thing hanging off the brain. The cerebellum, this is figure 15 showing the cerebellum. And the things on the cerebellum that you have to do in lab are the hemispheres. So it has sort of two distinct halves. It has hemispheres. It has the vermis, which is sort of a funny-looking structure down the midline. And it has the arbor vitae, that means tree of life. Vermis means worm. The arbor vitae, the tree of life, so it's not well illustrated here. It's the white matter inside the cerebellum branching out. And so these type of things you're going to see in lab on the cerebellum. And then the last part, oops, this should be at the same level as these two. Messed up with my bullets. So this should be at the same level as these two. The myelencephalon is the last part of the brainstem. And that's the part that's called medulla oblongata also. And that's where the brain blends into the spinal cord. So the brainstem has the three main parts: mesencephalon, metencephalon, and myelencephalon. And sorry, this is really just kind of giving you some general regions. So you can imagine there are courses you could take on the brain. right? So we don't have the time. We're running out of time in a serious way. So I'm not going to talk anymore about the brain. I didn't give you any functional stuff, really. There's going to be a lot more in the book. Even in your A&P book, we'll have more on these things. But we're just going to gloss over that, okay? So we'll just keep it to these regions, know the regions, okay? And next time, brainstem, medulla. Next time we'll start with the protections on the brain, and we'll pick it up with this, okay, on Wednesday. I still have quite a few. If any of you still need to pick up your exams, you can. Important.